Chapter 3 covers microscopy and staining. Alright, so we're going to start with the introduction. Come closer to an ordinary clean household pen, and what do you see? Probably not more than you saw from farther away. If you enjoy normal vision, you can see dust particles as small as 20 micrometers glimmering in a shaft of sunlight. However, what is amazing is how much you aren't seeing. Most bacteria are only half to 2 micrometers long and therefore remain invisible without the help of a microscope. With just a tenfold increase in visual acuity, you begin to see this busy, squirming microbial world. How might such an enhanced vision affect your desire to lift that clean fork to your mouth or your appetite for those clean vegetable sticks from the salad bar? How many bacteria can fit on a pinpoint? Look at the successive close-up pictures here and start counting. Each gold rod is an artificially colored bacterium. If you were to stick yourself with a clean pin, how many bacteria might be left behind in your finger? And how many are necessary to start an infection? Sometimes as few as 10 bacteria are enough. So you can see in the picture to the right, and this is on page 51 if you have your book, the close-up images of the tip here, starting with 91 times, they just look like gold dust. Moving to 455 times, we can start seeing those images, but going all the way up to the last picture, you can see how clear those rods actually are. Looking at historical microscopy, the picture here of Leeuwenhoek is on page 53. He was the first person to see individual microorganisms. He constructed simple microscopes capable of magnifying objects 100 to 300 times. These instruments were unlike what we commonly think of as a microscope today, consisting only of a single lens. They were very powerful magnifying glasses. But being that they were a single lens, it was very difficult to focus on one of his microscopes. So instead of changing the specimen, like moving the slide into the microscope, he basically just built a brand new microscope for each specimen, because once they were connected, it was very difficult to disconnect them. In a letter to the Royal Society of London in 1676, Leeuwenhoek described his first observations of bacteria and protozoa in water. He kept his techniques secret, however. He did not want to share any information about the microscopes he had created. Even today, we're not sure of his methods of illumination, although it is likely that he used indirect lighting, with light bouncing off the sides of specimens rather than passing directly through them. So the light was indirect. Leeuwenhoek was unwilling to part with any of the 419 microscopes he made. It was only near the time of his death that his daughter, under his direction, sent 100 of those microscopes to the Royal Society. Following his death in 1723, no one came forward to continue the work of perfecting the design and construction of the microscopes he created. Therefore, progress in the world of microbiology slowed tremendously. Through Leeuwenhoek's letters to the Royal Society in the mid-1670s, the existence of microbes was revealed to the scientific community. And then in 1683, he described bacteria taken from his own mouth. Leeuwenhoek could see very little detail of their structure, though, so further study required the development of complex microscopes, such as we soon shall see. All right, so principles of microscopy, we're still on page 53, looking at metric units. Microscopy, defined, is the technology of making very small things visible to the human eye. Because microorganisms are so small, the units used to measure them may be unfamiliar. So we're going to mention a couple here. Micrometer is equal to 0.000001 meter, or it can be expressed as 10 to the negative 6. And then the second unit, the nanometer, is expressed as 10 to the negative 9. And then we have the table there below, table 3.1, showing us our basic units that we should be familiar with: meter, centimeter, millimeter. Now we have our micrometer and our nanometer are two that we're going to add. And then if we look at the picture on the right, we can see the size of various things. Dog being 1 meter, tick being 10 millimeters, going to atoms, 0.1 nanometer. So that kind of gives us our relative size. Properties of light, wavelength and resolution. So this picture here of a wavelength is on page 54. Light has a number of properties that affect our ability to visualize objects, both with the unaided eye and with the microscope. Understanding these properties will allow you to improve your practice of microscopy. One of the most important properties of light is a wavelength, or the length of the light ray, represented by the Greek letter lambda. Wavelength is equal to the distance between two adjacent crests. We can see the crest on top, or two adjacent troughs. We can see the troughs on the bottom of a wave. So the green there is the wavelength. So we see the lines going up and down, up and down. So one wavelength constitutes from one crest to the next crest, or from one trough to the next trough, as those are equal. The sun produces a continuous spectrum of electromagnetic radiation with waves of various lengths. So we can see there on the increasing and decreasing wavelengths that we have various colors. The colors of the rainbow, Rory G. Biv, or red, orange, yellow, green, blue, indigo, and then violet. So the wavelengths there are increasing or decreasing based off of energy level and resolving power. So we see that we increase the wavelength as we go from gamma rays to radio waves, and we increase energy level and resolving power as we go from radio waves through gamma rays. So we see still that wavelength, but we notice there on gamma rays the crests are closer together, and on radio waves the crests are much farther apart. So the wavelength used for observation is crucially related to the resolution that can be obtained. Resolution defined then refers to the ability to see two items as separate and discrete units rather than a fuzzy overlapping single image. So we're looking on page 55, A versus B. In A, the two dots are resolved, that is, they can clearly be seen as separate structures. In B, these two dots are not resolved, therefore they appear to be fused. We can magnify objects, but if the objects cannot be resolved, then magnification is useless. Light must pass between two objects for them to be seen as separate things. If the wavelength of the light by which we see objects is too long to pass between them, then they will appear as one. So wavelength going down on the radio wave side, or the color red. The key to resolution is to get light of short enough wavelength to fit between the two objects we want to see separately. Cell structures less than 1/2 a wavelength will not be visible. Okay, still on page 55. Microscopists use shorter and shorter wavelengths of electromagnetic radiation to improve resolution. Visible light, which has an average wavelength of 550 nanometers, cannot resolve objects separated by less than 220 nanometers. Ultraviolet light, on the other hand, which has a wavelength of 100 to 400 nanometers, can resolve separations as small as 110 nanometers. Thus, microscopes that use ultraviolet light instead of visible light allowed researchers to find out more about the details of cellular structures. And then smaller objects corresponding to shorter wavelengths can pass more easily between the arms of the letter E, defining it more clearly. Therefore, we get a sharper image. So we see on the right here, if we were to take our objects and compare them to wavelengths, we know that the bigger the wavelength, the less resolution we have. The smaller the wavelength, the beads, the more resolution we have. Various things can happen to light as it travels through a medium such as air or water and strikes an object. So we'll look at how some of those things some of those things work, and consider how they can affect what we see through a microscope. So the first thing we're going to look at is reflection. If the light strikes an object and bounces back, giving the object color, we say that reflection has occurred. Light rays in the green range of the spectrum are reflected off surfaces of the leaves of plants. Those reflected rays are responsible for our seeing the leaves as green. So whatever color you see is the color being reflected. Transmission refers to the passage of light through an object. You cannot see through a rock because light cannot pass through it as it does through a glass window. In order for you to see objects through a microscope, light must either be reflected from the objects or light must be transmitted through them. Most of your observations of microscopes will make use of transmitted light. Absorption: if light rays neither pass through nor bounce off an object but are taken up by the object, then absorption has occurred. Energy in absorbed light rays can be used in various ways. For example, all wavelengths of the light's sun rays, except those in the green range, are absorbed by a leaf. Some of the energy in these other light rays is captured by photosynthesis and used by the plant to make food. Energy from absorbed light can also raise the temperature of an object. So a black object, which reflects no light, will gain much heat faster than a white object, which reflects all light rays. In some cases, absorbed light rays, especially UV light rays, are changed into longer wavelengths and re-emitted. This phenomenon is known as luminescence. So if the wavelengths are changed into longer wavelengths and then re-emitted back out, the object is said to be luminescent. If luminescence only occurs during irradiation, when the light rays are striking an object, the object is said to fluoresce. So minerals, gemstones, and various animals such as the green-eye fish and the pumpkin toadlets, which are frogs, fluoresce. If an object continues to emit light rays, if an object continues to emit light when light rays no longer strike it, then the object is phosphorescent. So some bacteria that live deep in the ocean are indeed phosphorescent. All right, so we're on page 56, figure 3.8, talking about refraction, which is defined as the bending of light as it passes from one medium to another of different density. The bending of the light rays gives rise to an angle of refraction, or the degree of bending. Look at the picture to the right. The underwater portion of the pencil seems to bend, but when you remove the object from the water, it is clearly straight. It appears to bend because the light rays deviate or bend when they pass from water into the air as their speed changes across the water-air interface. The index of refraction of a material is a measure of the speed at which light passes through a material. When two substances have different indices of refraction, light will bend as it passes from one material to another. So the refraction of light rays passing from water into air causes the pencil to appear to be bent. 57. Light passing through a glass microscope slide through air and then through a glass lens is refracted each time it goes from one medium to another. This causes loss of light and a blurred image. To avoid this problem, microscopists use immersion oil, which has the same index of refraction as glass, to replace the air. So when immersion oil is put onto the slide, then the light isn't refracted from immersion oil through to the slide. So a layer of oil joins the slide and the lens together. Thus, there is no refraction to cause the image to blur. Staining a specimen also increases differences in indices of refraction, making it easier to observe details. See the picture on the right? This trick works because light is not bent when it passes from one medium to another with the same index of refraction. So we can see the water there, the object in it, and in the immersion oil, since the indices of refraction are the same, we don't see the object through the water. Or through the immersion oil. All right, so properties of light. So this just sums up the definitions we went over. Reflection, being reflected back. Transmission, going through. Absorption, staying in the object and then re-emitting it back through if it fluoresces. And then refraction, looking or appearing as though it is bent. This example is on page 56, figure 3.7. All right, so looking at light microscopy, moving on to page 58, figure 3.11. Light microscopy refers to the use of any kind of microscope that uses visible light to make specimens observable. The modern light microscope is a descendant not of Leeuwenhoek's single lenses, but of Hooke's compound microscope, a microscope with more than one lens. Single lenses produce two problems. They cannot bring the entire field into focus simultaneously, and there are colored rings around objects in the field. Both problems are solved today by the use of multiple correcting lenses placed next to the primary magnifying lens. Used in the objectives and eyepieces of modern compound microscopes, correcting lenses give us nearly distortion-free images. See the object to the right? What we refer to as a single objective lens is really a series of several lenses which are necessary to correct aberrations of color and focus. The best objectives have as many as a dozen or more elements. So there are several lenses within each individual objective. Compound light microscope, picture on figure 3.12, page 58. The optical microscope or light microscope has undergone various improvements since Leeuwenhoek's time and essentially reached its current form shortly before the beginning of the 20th century. The microscope is a compound light microscope, that is, it has more than one lens. The parts of a compound microscope and the path light takes through it are shown in that figure. A compound microscope with a single lens is known as ocular. It's said to be monocular. One with two eyepieces is said to be binocular. Light enters a microscope from a source in the base and often passes through a blue filter which filters out the long wavelengths of light, leaving the shorter wavelengths to improve resolution. Remember, the shorter the wavelength, the better the resolution. It then goes through a condenser which converges the light beams so they pass through the specimen itself. The iris diaphragm controls the amount of light that passes through the specimen and into the objective lens. The higher the magnification, the greater the amount of light needed to see the specimen clearly. The objective lens magnifies the image before it passes through the body tube to the ocular lens in the eyepiece. The ocular lens then further magnifies the image. A mechanical stage allows precise control of moving the slide, which is especially useful in the study of microbes. So you can use the mechanical stage to move the slide left and right, up and down. The focusing mechanism consists of a coarse adjustment knob, so we see on the right, which changes the distance between the object lens and the specimen fairly rapidly. So that means that the slide that's sitting on the mechanical stage can move up towards the objective lenses or down very quickly by using the coarse adjustment. And then once you get it in focus, you use the fine adjustment knob to bring the specimen into sharp focus. So the fine adjustment knob is not to move the slide up and down, but to take something that's in focus and bring it into better or a sharper focus. Compound microscopes have up to 6 interchangeable objective lenses that have different powers of magnification. The microscopes that we have are going to have 4 objective lenses. And those are the 4 that we mentioned below. The total magnification of a light microscope is calculated by multiplying the magnifying power of the objective lens, the lens used to view your specimen, by the magnifying power of the ocular lens, the lens closest to your eye, so the eyepiece. Typical values for a microscope with a 10 times ocular lens, so our ocular lens are 10 times, are going to be for various objectives: the scanning objective, the low power objective, the high dry power objective, and then oil immersion. So remember, the eyepiece or the ocular lens is 10 times. So that's magnifying the specimen 10 times. And then whatever magnification you're looking at is going to multiply that number by the number in the ocular lens, which is 10. So the scanning objective, or the red objective, has a power of 3 times. So 3 times the 10 times in the ocular lens gives you a total magnification of 30. The low power, the yellow objective, has a 10 times scanning objective. And so 10 times the scanning objective, and that should be 10 times the ocular lens, gives you 100 times magnification. The high dry, the blue, gives you a 40 times magnification times the 10 times magnification in the ocular lens, gives us a total of 400 times magnification. And then the oil immersion, which is white, gives you 100 times magnification multiplied by the 10 times magnification in the ocular lens, gives us a total magnification of 1,000 times. So you may want to add these to your notes. The scanning objective is the red objective. The low power objective is the yellow objective, and that should say 10 times for the yellow objective and 10 times for the ocular lens, giving you a total magnification of 100 times. The high dry then is the blue objective, and the oil immersion is the white objective. And at the very end, after the equal sign, those are all total magnifications there. Looking at dark field microscopy, the condenser used in an ordinary light microscope causes light to be concentrated and transmitted directly through the specimen. This gives bright field illumination. So see figures 3.13a and 3.14a. And this is going to be on page 59. In some cases, however, it is more useful, especially with light-sensitive organisms, to examine specimens that would lack contrast with their background in a bright field under illumination. Live spirochetes, spiral-shaped bacteria that cause syphilis and other diseases, or organisms that would take that you would want to use that dark field microscopy for. A microscope adapted for dark field illumination has a condenser that prevents light from being transmitted through the specimen, but instead causes the light to reflect off the specimen at an angle. When these rays are gathered and focused into an image, a light object is seen on a dark background. So we can see a comparison of illumination in bright field and dark field microscopy. So bright field microscopy we see in A, where the light rays go through, directly through the specimen. In B, the dark field condenser deflects light rays so that they reflect off the specimen at an angle before they are collected and then focused on the image there at the bottom. All right, so the result then is going to be these two pictures: bright field illumination on the left, dark field illumination on the right. The dark field illumination is the one we were talking about that gives us an enormous increase in contrast because the slide is black. Therefore, the microorganisms are colorless or sometimes stained, and we can see those in a vast difference of contrast. Phase contrast microscopy. Most living organisms are difficult to examine because they cannot be stained by coloring them with dyes, because stains usually kill the organism. So to observe them alive and unstained requires the use of phase contrast microscopy. Light passing through objects of different refractive indices is slowed down and diffracted. This changes in the speed of the light are seen as different degrees of brightness. So the speed of light gives us the varying degrees of brightness, as we see here to the right in figure 3.15. That's our phase contrast image of an amoeba. Nomarski differential interface contrast microscopy. Like phase contrast microscopy, this one makes use of differences in the refractive index to visualize unstained cells and structures. However, this microscope produces a much higher resolution than the standard phase contrast microscope. So we can see the protozoa here, and the contrast is much higher, so the resolution is much better. Has a very short depth of field and can produce nearly a 3D image. So this one compared to the amoeba in figure 3.15, we can see how big of a difference the resolution actually is, and how this one gives us almost a 3D-like image. The next one is fluorescent microscopy. An ultraviolet light is used to excite molecules so that they release light of a longer wavelength than that originally striking them. The different wavelengths produced are often seen as various colors, brilliant shades of orange, yellow, or yellow-green, as we can see there to the right. This is figure 3.17. Fluorescent antibody staining is now widely used in diagnostic procedures to determine whether an antigen, a foreign substance such as a microbe, is present. Antibodies are molecules produced by the body as an immune response to an invading antigen, and antibodies are found in blood and serum. If the patient's specimen contains a particular antigen, the antigen and antibodies specifically made against it will kind of stick together. So when they clump together, we want to see those reactions occurring, and that reaction is ordinarily not visible. But if fluorescent dye tagged antibodies against syphilis organisms are added to a specimen containing spirochetes, when the antibodies, since they're tagged with the fluorescent dye, bind to an antigen, then they give off a color. And then those organisms can be identified as the cause of syphilis. Diagnosis can be made in minutes rather than in hours or days it would take to isolate, culture, and then identify those organisms. So we can just tag an antibody, put the antibody in. If the antibody binds to the antigen, the antibody lights up, and we know that it has bound to that particular antigen. Digital microscopy, this one's pretty cool. This guy is on page 62. Have you had frustrating moments in the lab when you just couldn't get a slide into focus? Then you would like the autofocus, auto aperture, auto light, motorized stage, and magnification changers of a digital microscope. Not only that, but these microscopes also come with built-in camera and preloaded software. All you have to do is plug in the unit, turn on the power, use the mouse to view live or stained specimens on a monitor, so you don't even have to focus the slide. There are some limitations. Maximum magnification is limited, and they are extremely pricey. Moving on to another advanced microscope, an electron microscope, page 63, figure 3.21. The light microscope opened doors to the world of microbes. However, because it could not resolve objects separated by less than 0.2 micrometers, the view was limited to observations at the level of the whole cells and their arrangement. So we could see, like we did in the beginning of the picture, rods or bacilli. We see the whole cell, and we can see how they arrange together. But few subcellular structures could be seen, and we can't see viruses with these compound microscopes either. So the advent of the electron microscope, EM, allowed these structures, these very small structures, to be visualized and thus studied. The EM uses a beam of electrons instead of a beam of light, and electromagnets rather than glass lenses to focus the beam. The electrons must travel through a vacuum because collisions with air molecules would scatter the electrons and result in a distorted image. And you can see the electron microscope to the right. I worked with one in particular, a scanning electron microscope, when I took a class at Eastern. It was an SEM class, scanning electron microscopy, and those microscopes are extremely complex and advanced. So we can see that microscope there in the picture at the bottom on the right. Electron microscopes are much more expensive than light microscopes. They take up much more space and require additional rooms for preparation of specimens and processing of photographs. The two most common types of electron microscopes are the transmission electron microscope, the TEM, and the scanning electron microscope, the SEM. Both are used to study various life forms, including microbes. The more advanced scanning tunneling microscope and atomic force microscope let us see actual molecules and even individual atoms. So we can see the picture with the TEM versus the picture with the SEM. Moving on to techniques of light microscopy. So we're going to page 68. Microscopes are of little use unless the specimens for viewing are prepared properly. Here we will explain some important techniques used in light microscopy. Although resolution and magnification are important in light microscopy, the degree of contrast between structures to be observed and their background is equally important. Nothing can be seen without contrast, so special techniques have been developed to enhance contrast. So the first technique is the wet mount. Wet mounts in which a drop of medium containing the organism is placed on a microscope slide can be used to view living organisms. We will do wet mounts throughout the semester. Smears in which microorganisms from a loop full of medium are spread onto the surface of a glass slide can be used to view killed organisms, and we'll do smears as well. Although they are living when placed on the slide, the organisms are killed by the techniques used to fix or attach them to the slide. So once we do a smear, we will do a heat fix to kill the organism. Heat kills organisms, as well as attach the microorganism to the slide. Smear preparation is often difficult for beginners. If your smears are too thick, you will have trouble seeing individual cells because you'll have too many on the slide. And if you make them too thin, then you may not find any microorganisms on the slide at all. So you have to get an equal medium, and no worries, we will practice this technique extensively in class. If you stir the drop of medium too much as you spread it on the slide, you will disrupt cell arrangements. So you'll break those cells up, and you won't be able to see the arrangements they appear in. Like sometimes cells will arrange in tetrads or in diplo or in strepto or in staphylo. So if you mix them too much, you're going to disrupt those arrangements and you won't be able to see how they arrange together. And that will come in handy at the very end when we do the dichotomous puzzle and we try to identify the genus and species, because various species tend to have differing arrangements. Such variations lead some beginners to imagine that they see more than one kind of organism when in fact the organisms are all the same species. So if you disrupt those arrangements, you may see some staphylo and some diplo and some tetrad. So you may think that you have more than one species when actually you just did your smear incorrectly. After a smear is made, allow it to dry completely, air dry completely. It has to air dry completely before you heat fix it. If it is not completely dry when you heat fix it, the organisms will be boiled and destroyed. Some of them can get into the air. So sometimes when you heat fix a slide that is still wet, the microorganisms can get in the air. If they're still living and you breathe those in, those can make you sick. So heat fixation accomplishes three things: kills the organism, makes it safe to work with, causes the organism to adhere or stick to the slide, so that way these organisms are readily available to accept the stains or the dyes that we put on next. Keep in mind though that it has to air dry completely before you heat fix it so we don't destroy the microorganisms, and also so we don't make those microorganisms aerosolic. If you heat fix too little, the organisms may not stick, and then they'll wash off the slide whenever you rinse the slide off with water and stains. And the cells that remain alive, they won't be readily available to accept the stain, so they'll stain very poorly. But if you heat fix too much, the organisms will be incinerated, and when you look under the microscope, you'll see black specks, so you'll see distorted cells and cellular remains. So there is a happy medium with heat fixing as well. You want to run the slide through the Bunsen burner 2 to 3 times to make sure that you properly heat fix, kill, and fix the microorganisms to the slide. A stain or dye is a molecule that can bind to a cellular structure and give it color. Staining techniques make the microorganisms stand out against their backgrounds. They are also used to help investigators group major categories of microorganisms, examine the structural and chemical differences in their cellular structures, and look at the parts of a cell. In microbiology, the most commonly used dyes are cationic. Cations are positively charged. These are known as basic dyes, such as methylene blue, crystal violet, safranin, and malachite green. These dyes are attracted to any negatively charged cell components. The cell membranes of most bacteria have a negatively charged surface and thus attract the positively charged basic dyes. So that's how they're able to stick together. Other stains such as eosin and picric acid are anionic or negatively charged. These are acidic dyes. They are attracted to positively charged cell materials. Two main types of stains, simple stains and differential stains, are used in microbiology, and we'll be doing both of those, and they're compared in table 3.3, and this is going to be on page 71. A simple stain makes use of a single dye and reveals basic shapes and cell arrangements. Methylene blue, safranin, Carmel fusion, and crystal violet are commonly used simple stains. So when you put the simple stain, you're only using one stain, so all of the cells stain just one color. Now, if you had more than one specimen, more than one species there, then they would all stain that color, so you wouldn't be able to differentiate between something that was like gram-positive and gram-negative. The simple stain is just basically telling you that there are bacteria or there are organisms present. On the other hand, a differential stain makes use of two or more dyes and distinguishes between two types of organisms or between two different parts of an organism. So common differential stains are the gram stain, which tells us we have gram-positive or gram-negative bacteria. We'll be doing that guy. The Ziehl-Neelsen acid-fast stain and the Schaefer-Fulton spore stain. We will also be doing a spore stain. So to start out with on this differential stain, the gram stain, probably the most frequently used differential stain was devised by Danish physician Hans Christian Gram. That should be Gram, G-R-A-M. In 1884, Gram was testing the new methods of staining biopsy and autopsy materials, and he noticed that there were certain methods of some bacteria that were stained differently than the surrounding tissues. As a result of his experiment with stains, the highly useful Gram stain was developed. In Gram staining, bacterial stains take up all crystal violet. So crystal violet is the first stain to be added, and then iodine is added. Iodine acts as a mordant, so it allows crystal violet to be fixed inside of the cells. So all the cells look red. All the cells look purple. And the cells look purple because of some varying characteristics in the cell. In gram-positive bacteria, they have up to 40 layers of peptidoglycan. So the iodine is going to fix the crystal violet in those layers of peptidoglycan permanently. Now, when we decolorize with alcohol, our gram-negative cells only have about 2 to 3 layers of peptidoglycan, so the crystal violet isn't going to fix permanently in the 2 to 3 layers of peptidoglycan. So alcohol acts as the decolorizer, and it decolorizes all of the gram-negative cells. So the 40 layers of peptidoglycan are still going to hold on to purple, so the gram-positive cells are going to remain purple. But the gram-negative cells, since they only have 2 to 3 layers of peptidoglycan, they're not going to hold on to that crystal violet. So now we have just decolorized
only the gram-negative cells. So when we put safranin on there, safranin is red, and safranin likes to stick to fats and lipids, and that's what comprises gram-negative bacteria, but we don't see those in gram-positive bacteria. So now those clear gram-negative cells are going to hold on to safranin, those lipids are going to hold on to safranin, and so now the gram-negative cells are going to appear red or pink. So when we look into the microscope, the gram-positive cells are going to remain purple, gram-negative cells are going to remain red or pink. So here's a picture of a gram stain to the right. 4 groups of organisms can be distinguished with a gram stain. Gram-positive organisms whose cell retain crystal violet stain, gram-negative organisms whose cell do not retain crystal violet stain, remember those are going to be red. Gram non-reactive organisms which do not stain or stain very poorly, gram variable organisms which stain unevenly. The differentiation between gram-positive and gram-negative cells reveals a fundamental difference in the nature of the cell walls. So as I mentioned earlier, the peptidoglycan versus the lipids, and we will learn more specifically about those cell structures in chapter 4. The Ziehl-Neelsen acid-fast stain, this is on page 70, figure 3.31. This stain can be used to detect tuberculosis and leprosy-causing organisms of the genus Mycobacterium. Slides of organisms are covered with chiral fusion and are heated, rinsed, and decolorized with 3% hydrochloric acid and 95% ethanol. Then it's rinsed again and stained with Loeffler's methylene blue. Most genera of bacteria will lose the red chiral fusion stain with the decolorizing step, but those that are acid-fast retain the bright red color due to, again, the lipids in their cell wall. So we can see then if they are bright red, then they are acid-fast bacteria. Negative stains, and we will complete a negative stain this semester. They're used when a specimen or part of it, such as the capsule, resists taking up the stain. The capsule is a layer of polysaccharide material that surrounds many bacterial cells and can act as a barrier to host defense mechanisms. So very virulent, very strong, very pathogenic bacteria tend to have capsules. Capsules makes it very difficult to kill those particular bacterial strains. So in negative staining, the background around the organism is filled with a stain such as nigrosin. So we see that nigrosin is covering the entire slide. It looks purple, but it's actually black. This process leaves the organisms themselves as clear, unstained objects that stand out against the dark background. A second simple or differential stain can be used to demonstrate the presence of the cell inside the capsule. Thus, a typical slide will show a dark background, which looks purple here, and a clear unstained area of the capsule. So the clear zones around the rods are the capsules. So what they did was they stained the slide, they left the cells colorless, but then they added another stain into the cells. So that's why the rods are stained, but the capsule doesn't take up the stain, so the capsule is clear itself. We'll be doing a basic negative stain on bacteria that do not contain capsules because they're pretty virulent. So what we will see is a negative background, a black background, and the cell itself is going to remain clear rather than the capsule that we're looking at right there. And this negative staining is, again, on page 70, figure 3.32. Now, flagella staining. Flagella are appendages that some cells have and use for locomotion and are too thick to be seen easily with a light microscope. When it is necessary to determine their presence or arrangement, flagellar stains are painstakingly prepared to coat the surfaces of flagella. So we can complete a stain that coats the flagella that allows us to see those flagella as well. And those stains are usually with a dye or metal such as silver. Endospore staining, one that we will be doing this semester, page 72, figure 3.33. A few types of bacteria produce resistant cells called endospores. Endospore walls are very resistant to penetration of ordinary stains. When a simple stain is used, the spores will be seen as clear, glassy easily recognizable areas within the bacterial cell. Thus, it is not necessary to perform an endospore stain, but the differential Schaefer-Fulton spore stain does make spores easier to visualize, and that's because it dyes the spores green. Heat-fixed smears are covered with malachite green and then gently heated until they steam. So we have to use a beaker with water and steam the glass slide, and that's because the dye does not easily penetrate the spore, so a gentle heat steam allows the dye to readily get inside the endospore much faster. So we use the steam for about 5 minutes, causing the endospore walls to become permeable to that malachite green. Then the slide is rinsed with water and for 30 seconds to remove the dye from everywhere except for inside the spore. So we can't rinse the green out of the spore, but we do rinse it out of the cell itself. So then we counter stain the cell with safranin. Whether it's gram-positive or negative, that doesn't matter. Safranin is going to go into these cells, and so it's going to make the cells appear red. So all cells, whether they were gram-positive or gram-negative, are going to appear red. And then if there's an endospore inside of that cell, that endospore is going to appear green. Although microscopy and staining techniques can offer valuable information about microorganisms, these methods are usually not enough to permit identification of most microbes. Many species look identical under the microscope. After all, there are only a limited number of basic shapes, arrangements, and staining reactions, but thousands of bacteria. This means that biochemical and genetic characteristics usually must be determined before identification can be made. We will, however, towards the end of the semester, be completing a dichotomous puzzle, a two-part process in which you're going to utilize various stains that we talked about today, such as a gram stain and the endospore stain. And with a dichotomous puzzle, I will have specific types of genus, species, names of bacteria that are characterized by these varying stains, so you will be able to identify a particular genus, species, name of a bacterium based off of all of the lab techniques that we do throughout the semester.